Hey everyone, and welcome to today's Explainer. I am absolutely thrilled to guide you through this high-yields whiteboard journey. For the graduate learners tuning in, you know that really grasping these fundamental pathways of disease is absolutely crucial. So today, we're going to track the life of a single cell. We'll follow its entire journey from a state of perfect, happy balance, homeostasis, all the way through adaptation, injury, and ultimately death. We are going to visualize exactly what goes wrong at both the molecular and the structural levels. Let's get into it. So I want you to think about this for a second. What actually happens the exact moment a cell gets pushed past its limit? Because, cells operate within a very narrow, highly regulated set of constraints. But the environment inside the human body, it's constantly changing. When massive stress hits, whether that's from structural damage, fluid imbalances, genetic mutations, or just aging, the cell has to make a choice. It must either adapt to that stress or face irreversible injury. The stakes literally couldn't be higher for our cellular survival. Okay, let's dive into this. Here is our clear pathophysiologic flowchart for today. Cellular adaptation to stress, mechanisms of cellular injury, the hypoxic cascade, accumulations and pathologic calcification, and we'll wrap up with cellular death. Starting right off with Section 1, cellular adaptation to stress. Because a stressed cell isn't immediately an injured cell, its very first survival instinct is to adapt. And this brilliantly illustrates that concept. A cell will actually alter its size, its number, or its phenotype to handle whatever physiologic demands or pathologic conditions are being thrown at it. If you look at a normal, simple cuboidal epithelial cell, you can see how radically it changes its physical architecture in response to different stressors. This adapted cell is kind of living in a functional gray area. It's not completely normal anymore, but it's not truly injured yet either. It's just doing whatever it takes to survive its environment. Let's hit some rapid-fire definitions here, starting with size. Atrophy is a decrease in cell size. If you put a broken limb in a cast, those skeletal muscle cells shrink from disuse. They literally degrade their own proteins to conserve energy. Now, hypertrophy is the exact opposite. It's an increase in cell size. A classic, heavily tested example of this is left ventricular hypertrophy. When the heart has to pump against chronic high blood pressure, those myocardial cells synthesize way more proteins and physically expand to handle the heavy lifting. Both of these are totally reversible survival tactics, depending on the workload. Moving on to cellular numbers and types. Hyperplasia is an increase in the actual number of cells driven by cellular division. Think of your liver rapidly regenerating after a partial resection. Metaplasia is fascinating. It's the reversible replacement of one mature cell type with another. Like in chronic smokers, those fragile ciliated columnar cells in the lungs, they get replaced by tougher, stratified squamous cells just to withstand the smoke. But be careful with dysplasia. Dysplasia is deranged, completely atypical, disorganized growth. While it kind of sounds related to hyperplasia, dysplasia is absolutely not a true adaptive change. It's a massive red flag that can precursor invasive tumors if that stressor isn't removed. All right, moving into Section 2. Mechanisms of cellular injury. We are now crossing the threshold where adaptation fails. The stress has become way too severe, or maybe it's just gone on too long. An active cellular injury begins. Regardless of whether that cell is getting injured by a chemical toxin, a bad infection, or physical trauma, the biochemical themes of injury are remarkably universal. We always see profound ATP depletion. We see the dangerous accumulation of reactive oxygen species, causing massive oxidative stress. We see the sudden, catastrophic entry of calcium ions right into the cytosol. And finally, mitochondrial membrane damage alongside severe protein misfolding. These four specific events are the exact triggers that compromise the cell's plasma membrane and its internal organelles. Which brings us to Section 3, the hypoxic cascade. Hypoxia, a lack of sufficient oxygen within cells, is hands down the single most common cause of cellular injury. If you're in a graduate pathophysiology course, you already know this exact sequence is heavily tested. Let's walk through this cause and effect pathway. The most common cause of hypoxia is ischemia, meaning a reduced blood supply, like from a blood clot. No blood means oxygen delivery to the mitochondria totally drops. Without oxygen, those mitochondria can't make ATP. Now here's the so what. Your cell's sodium-potassium pump relies entirely on that ATP to push sodium out. When the pump fails, sodium builds up inside, and in biology, where sodium goes, water inevitably rushes in right behind it. The entire cell rapidly swells up with water, a process called oncosis. Let's move along and see how this builds. To survive without oxygen, the cell desperately resorts to anaerobic glycolysis to generate whatever tiny bit of energy it can. but doing this consumes all its glycogen stores and produces lactate, or lactic acid. As that lactic acid builds up, the intracellular pH drops precipitously. This new, highly acidic environment is terrible for the cell. It actually causes the nuclear chromatin to clump together, which severely disrupts the cell's genetic machinery. So the crucial point here is understanding what pushes a reversibly injured cell way past the point of no return. And the culprit? It's calcium. Normally, calcium is kept at extremely low levels in the cytosol, but during severe injury, calcium just pours in from the outside and leaks out from those damaged mitochondria. This unbuffered free calcium acts like an internal wrecking ball. It uncontrollably activates destructive enzymes, phospholipidases that chew up the cell membrane, proteases that completely dismantle the cytoskeleton, and endonucleases that fragment the nucleus. Once these get activated, severe injury is, actually, scratch that. Full-on cell death is absolutely guaranteed at that point. Now we encounter a fascinating biological irony, the paradox of reperfusion injury. If a cell is dying from ischemia, the obvious, common-sense treatment is to restore blood flow and reoxygenate the tissue, right? While restoring oxygen to an already highly stressed, ischemic, and swollen cell can actually trigger immediate, massive necrosis. You're providing the very thing the cell needs to live, and it ends up destroying the whole shebang. You might be wondering, why on earth does this happen? Well, here are the exact mechanisms that make reperfusion lethal. And remember, this paradox is heavily tested. First, suddenly reintroducing oxygen induces massive oxidative stress, creating highly reactive oxygen and nitrogen radicals that just shred the already compromised cell membranes. Second, it drives even more of that lethal calcium into the damaged mitochondria, halting ATP production completely. Third, dying cells triggers cytokine danger signals, calling in an intense localized inflammatory response. And finally, the complement system activates, which just exacerbates the damage even further. Let's shift gears to Section 4, Accumulations and Pathologic Calcification. Now, what's really interesting about this is seeing what happens when stressed cells fail to process substances normally. Basically, they turn into cellular hoarders. These infiltrations can happen in the cytoplasm or the nucleus, and they can be incredibly toxic. This hoarding happens in 4 primary ways. First, abnormal metabolism can cause normal stuff, like fat, to build up. Hello, fatty liver disease. Second, a genetic mutation might cause proteins to misfold and stack up. Third, missing a vital lysosomal enzyme means complex substrates just can't be broken down, leading to devastating lysosomal storage diseases. And fourth, the cell might ingest exogenous in cereals it literally can't digest, like heavy metals or mineral dust, and they just sit there permanently. And speaking of hoarding, one of the most important accumulations to understand is pathologic calcification. Examiners love to test you on the difference between these 2 types. Dystrophic calcification occurs in dying or already necrotic tissues. Think of advanced atherosclerosis, or a damaged heart valve that gets all stiff and calcified, even though the patient has totally normal blood calcium levels. Metastatic calcification is an entirely different beast. It happens in perfectly normal, undamaged cells simply because the patient has systemic hypercalcemia, meaning there's too much calcium circulating in their blood, which then deposits right into healthy tissues. Finally, we arrive at our last stop, Section 5, Cellular Death, Necrosis versus Apoptosis. When irreversible injury occurs, the cell's journey is over, but how it ends matters immensely to the tissues around it. Use this high-yield table to really lock in the differences between the 2 main types of cellular death. Necrosis is a messy, inflammatory explosion. The cell swells up, its plasma membrane completely ruptures, and all its digestive enzymes leak out, damaging everything nearby and triggering heavy inflammation. Necrosis is always, invariably, a pathologic process. Apoptosis, on the other hand, is a neat, programmed cellular suicide. The cell actively shrinks, packages its contents into neat little apoptide bodies with perfectly intact membranes, and is quietly cleared away by macrophages without sparking any inflammation. Apoptosis happens all the time normally to get rid of unwanted cells, though it can also be triggered pathologically by severe DNA damage. To wrap things up, I want to leave you with this really profound quote regarding a third cellular survival process called autophagy, which literally translates to self-eating. When starved, a cell initiates cannibalization of its own organelles to recycle the parts and survive. But as the text notes, autophagy declines and becomes less efficient as cells age, a dynamic which contributes to the aging process. It really leaves us pondering this delicate, inevitable balance. The very mechanisms that allow our cells to adapt, survive, and recycle their own components slowly lose their efficiency over time, ultimately driving the biologic aging process we all experience. Which leaves us with a critical question for the future of medicine. If we can eventually learn to therapeutically control and prolong autophagy, could we one day hit pause on cellular aging entirely? Think about that. Thanks for joining me on this explainer.